Hi, my name is Lauren, and today I will be presenting our research from the Goel lab titled Sensory Processing Impairment is Correlated with Social Cognition Deficits in a Mouse Model of Fragile X Syndrome. For some context, Fragile X syndrome is the most common inherited form of intellectual disability and as well as the leading cause of autism. Some symptoms include hyperactive behavior, learning impairments, and other cognitive deficits. Recent studies suggest that sensory issues are associated with and even predictive of social deficits, and also that social interaction wild-type mice are influenced by olfactory, somatosensory, auditory, and visual cues. So we hypothesize that the impairment of specific sensory processing can impair social cognition. To test this hypothesis, we used young adult wild-type mice and FMR1 knockout mice, the most well-studied Fragile X syndrome mouse model. They were water-restricted and trained on a go, no-go visual task where they learned to lick preferably to a 45-degree orientation and withhold licking to a 135-degree orientation. After reaching expert status, mice are introduced to the distractor task. This includes trials with sensory distractors consisting of distracting sounds and lights. Now, to examine the social cognitive impairments, the FMR1 knockout mice performed a three-chamber sociability and social novelty task. During this task, the entire box is recorded, and we use these movies to train a deep learning network to analyze the interactions of the experimental mouse in the apparatus. This picture shows a demonstration of the visual discrimination task. As you can see, a mouse is running on a ball, and there's a lick port that allows the mouse to lick. In front of the mouse is a screen that presents different visual stimuli. In the second picture shows a similar setup, but now there are these distracting sounds and lights. In this third video, it shows the three-chamber task. The experimental mouse is placed in the middle chamber and is allowed to explore the box. For the sociability test, the outer chambers contain an upside-down cup, with one of them empty and the other with a stranger mouse, M1. During the social novelty task, the empty cup contained a novel mouse, M2, while the M1 was now the familiar mouse. In the first three plots, the y-axis shows the discriminability index, or D prime, which is the ratio of hits and correct rejections. And in the first plot, the visual task, the knockout mice took more training sessions to reach a D prime of 2, which is considered a very high level of learned performance, compared to the wild-type mice. Now, it's important to note that knockout mice still learn, but they just take longer compared to the wild-type mice. In the second and third plots, preliminary data shows that wild-type mice continue to perform well in distractor trials, but task performance in knockout mice drops significantly, which is indicative of hypersensitivity. Now, in the sociability plot, our preliminary data shows knockout mice exhibit elevated velocity compared to wild-type mice. And we look at velocity to get a sense of how much the mouse is moving around the cage versus spending time interacting with the mouse. And in the social novelty task, we see reduced interaction between the knockout mice and the novel mouse. So in this first plot, we see a negative correlation between social interaction time and the number of sessions it takes to reach a D prime greater than 2 for the normal task. The longer the interaction time, the less amount of sessions. And in the second plot, in the auditory distractor task, we see a positive correlation between social interaction time and discriminability index. The shorter the interaction time, the smaller the D prime. Now for the bottom two plots, we use the velocity data. The first plot shows a positive correlation between velocity and the number of days it takes to reach a D prime greater than 2. So in this plot, elevated velocity indicates that the mouse took more days to reach a D prime greater than 2. And this is what we would expect looking at the last plots in the previous slide where knockout mice showed elevated velocity. In the last plot, we see a negative correlation between velocity and social interaction time, meaning elevated velocity indicates shorter interaction time just for this sample. And again, this is all just preliminary data. So our data shows that during the sociability task compared to the wild-type mice, FMR1 knockout mice showed an elevation in velocity, indicating faster random movements and less time spent interacting with M1. We also found a positive correlation between the sensory discrimination deficits and social impairments. And our data suggests that similar to humans with Fragile X syndrome, sensory impairments in knockout mice are accompanied by deficits in social cognition, and therefore our experimental paradigms provides a powerful translational model to look at the sensory and social issues in human populations. And our next goal is to investigate the neural mechanisms that contribute to these social cognitive deficits. And thank you so much for watching.